Bismillah ar-Rahman ar-Rahim. Hello everyone, this is solving past paper 3-1 May-June 2018. Now the goal of this experiment is to find the concentration of unknown protein solution. And in order to do that, we simply need to make a standardized test. Now in order to make a standardized test, we need to start with a dilution or a simple dilution in order to make a range of concentration of the known concentration, and we're going to start with the stock solution provided from the examiner. The stock solution is 1% milk, and we need to make a different concentration that reduces the concentration by 0.2 every time. So here's an example about how we can prepare 0.8%, and in order to do that, simply you need to follow the formula, which is C1V1 equals C2V2, while the concentration 1 is the stock solution. The V1 is the unknown, which is basically the amount of the solution that you're going to take it from the stock. C2 is 0.8 because the stock is 1 and you need to reduce it by 0.2. So C2 for the first concentration is 0.8. And the V2 is 10 ml. You might wonder, how did we know that the V2 is 10 ml? Simply, if you look at the exam paper, you will see that there is a table provided for the concentration saying that the table has a milk concentration of 10 ml. So you can conclude that the V2 is 10 ml. So after you do the math, V1 gonna be 8 ml, 8 ml out of the stock solution, and you add 2 ml of water in order to make it 10 centimeter cubic of 0.8. So here's an example about how to make the dilution. Now in order to make the other concentration, you need to follow the same exact formula. After you do that, you need to, after you're doing the dilution, you need to start doing the experiment. Now let's look at the experiment. The experimental tools provided by the examiner. Basically, you are provided with the milk as well as you are provided with the unknown. The unknown is labeled U. Here's the unknown, and here's the stock solution. What we need to do is basically to find the concentration of unknown solution, and here's the stock solution by which the student are able to make the dilution. If you look at the test tubes right here, our student here, Tala, she made a dilution of the different concentration, but she forgot to label the test tubes. She should label the test tube so any mistake would not happen in the exam. So which one is the stock solution?
This one.
This one is 1%.
I ordered them. This is 1, this is 0.8, 0.6, 0.4, 0.2, 0.1.
And the 0.1. So we have 6 different concentration. Now after you make those 6 different concentration, what you need to do is to use a spotting tile. A spotting tile right here, she uses the spotting tile to drop 1 or 2 drops of the different concentration. So you pick any one of them and then you put it in the spotting tile. Before you put it in any wells of those spotting tile, the spotting tile should be labeled. So you add 1 or 2 drops of milk. After you add milk, as the examiner asks you to do, you need to add solution K, 1 or 2 drops also. After that, you add 0.5 centimeter cubic of C. What is C? C is basically a copper sulfate. Okay, it is stated in the cover page of the exam that C is a copper sulfate. So after you add 0.5 ml of copper sulfate, the color of the solution in every well will change. Now, as you can see, there are different colors for every one of those different concentration, and that's normal. Why it's normal? Because basically we are making a standardized test. Whenever we make a standardized test, that means we are doing a standard that we are going to compare the unknown with. So right here, the standardized test is about the concentration and color. So as you can see, here is the results for Tala. Can I see the well? Thank you very much. So the concentration 1, 0.8, 0.6, 0.4, 2, 1, and here's the unknown. Now Tala, she's doing the unknown. Let's see how the color of the unknown would look like. She placed the milk, 1 or 2 drops. How many drops of milk you add?
2 drops.
2 drops of milk. Now, once you add 2 drops of milk, for one of them, you need to add 2 drops for all of them, right? Because that's a controlled variable. After you added the milk, you added the K. How many drops of K you added?
Also 2.
2 drops. And finally, you add 0.5 ml of C. And here's the color of U. Now, what do you think, guys? We need to wait for 2 minutes, right?
Yeah, here.
We need to wait for 2 minutes before we judge. Tala, here's a question.
Yes.
It's not pop quiz. Now, do you think that there is a significant source of error once you are estimating the concentration of U?
Well, there could be human error because you don't actually have a measuring standard. You just compare with your eyes, maybe.
Okay.
And maybe these are placed for longer than the other one.
This is
So
very.
there is time variable.
Actually, there are 2 main sources of error. Correct. The first one is the misjudging color, as you said, and the second one, how would you abbreviate it? You said different concentration had different time, right? So once you started with concentration 1, you started with the 0.8, and after you finish the 0.1, you start timing 2 minutes. That means one took longer time compared to others. Do you know what does that mean? How is that stated? How that source of error can be, let's say, written or abbreviated? Simply, you can say different timing. Different timing. How would you improve the different timing? Simply by doing every solution separately. That's how would you avoid different timing. Let's look at Raneem's. Okay. As you can see, the 1, the 0.8, 0.64, 0.2, and 1. Now we need to compare those colors with the key provided by the examiner. After you compare it, you need to record your results in a table. The table should be ruled with a ruler, and you need to make sure that the independent is on the left side while the dependent on the right side, and basically the unit is written. Here is percentage, that's right. You wrote percentage, so you add the unit in the table. Well done, guys. And here's another guy right here. He's doing his experiment and he's doing his table carefully. You need to make your table carefully. It should be neat, nice. You can use pencil for making tables. He's done with the dilution, and he's done with the different concentration. He made a standard. 1, 0.8, 0.64, 0.2, and 0.1. Okay. Now, what can you tell about this color? Is it violet, pale violet? Violet. What about this one?
Violet.
What about this one?
Violet.
Violet. I think there is something
Pale
wrong
violet.
right here. Now, it's better to open the photo on your mobile so you can compare the results. Okay. I think there is something wrong right here because it should be a little bit lighter. Maybe you had something wrong with the dilutions.
ممكن extra concentration.
Is it violet or pale violet?
لا pale violet.
Pale violet.
High blue.
Now again, this is a source of error because it is very hard to judge what color is that. People see colors in different way, and that's one of the sources of error. How we can improve that source of error, Ahmed?
White tile.
White tile. Basically, it is on a white. It is a white tile.
Use a color meter.
Color meter. Thank you very much. Color meter is a device that is used to give quantitative results instead of qualitative. Quantitative instead of qualitative. Okay, you still need to do it with U and to judge what is the concentration of U. Okay then, so the unknown solution, if you do the unknown solution, you would have a specific color, and after you do the unknown solution, you need to compare the unknown solution with the other concentration in order to estimate the concentration of the unknown solution. That's the idea of standardized test. Now, according to your results, Raneem, you had the U of this color, and you recorded you as violet, right?
Yes.
And you compared you with the other concentration. After that, you estimated that U is 0. What do you think?
6.
You estimated that U is 0.6. Now that's based on your results. That's correct based on your results. Okay, so because now your grades would depend on your results. Okay, here is another estimation. Here's U with the, what did you estimate U?
0.8.
0.8 violet color, and here is a violet color. Based on your results, that's also correct. Again, as I said, there are many sources of error or significant sources of error right here. Different timing, misjudging color, as well as the volume or the size of the drop. The size of the drop may differ between every well, okay, and may differ between different students, so that's also a source of error. How can you improve this type of error?
El dropper.
Yeah,
Use a burette or a pipette.
a syringe or any measuring tool that measures volume instead of using the pipette. Okay, I think this is the end of this procedure, and after you do your results, you need basically to put it in a table, appropriate table that has the concentration and the color, and to find out the concentration of unknown solution. This is the end of the first procedure in this paper. There are two procedures actually. This is the first one, and there is another one which you need to follow in order to find out the agglutination in milk. Okay, now for the second procedure in this paper, you need to basically add hydrochloric acid for each one of those different concentration to test the agglutination process that takes place in milk. You know, guys, that milk has a protein, and that protein clots together whenever the pH becomes less. Now, in the natural way of making cheese, we place bacteria, which actually respires anaerobically and produces acid. And after the acid is produced, the milk, the protein in the milk clots, and then we have the cheese after we remove the water. Now, this process can be mimicked basically by adding acid, which is hydrochloric acid, into the different concentration of milk. Now here, we are making a standardized test, by the way, in order to see the agglutination in different concentration. Again, we are adding acid to different concentration in order to see the agglutination. And after we are done with those 6 different concentrations, we repeat the experiment with the unknown to find out the degree of agglutination for the unknown, and by doing that we can estimate the concentration of the unknown. So if you add the acid into different concentration, and apparently this one had more clotting compared to this one, and if you compare it with the diluted concentration right here, you will see this one is also had less clotting. Okay, so what you need to do, this one for example, it has very, or actually I cannot detect any clotting right here. It is, I think, the least concentration. You can record it as 1+. After you do that,